Maybe we can start with the introductions.
Mhm.
So thank you so much for joining us today, everyone. We have the wonderful Sergio here, who is a scientist at Columbia University, and we can get started. Sergio, do you want to tell a little bit about yourself, where you're from?
Yeah, sure. Thanks, Praching. So I'm originally from New York. Been in New York almost my entire life, and now I'm a graduate student at Columbia University. And I'm in my second year, so still relatively early in the graduate school career. And I joined a lab that focuses on neurobiology. Yeah.
That's wonderful. What got you into science in the first place?
Yeah, great question. So I think what really attracted me to science was just it really allows you to be curious and ask any question that you want. And you can really try and answer that question in a way. So you can come up with a question which we term, right, a hypothesis, and then you just find ways to test it and you find out sort of, are you right or are you wrong? And neither one, you can be right or you can be wrong, but both sort of bring you satisfaction in terms of, you find out something a little bit more, you learn in knowledge about that one subject area. Yeah.
That's so great. And what is your question or your hypothesis that you're working at?
Yeah, so specifically in my lab, we study the connections between neurons, which we call synapses. So we really focus on that connection between one neuron and the next, and I'll talk about it a lot in my presentation. But we're trying to understand what's the implication of having those connections, what can go wrong when you don't have enough of those connections. And it seems like humans, compared to any other animal that we can look at, have the most number of connections in their brain, despite our brain not being the biggest when compared to, for example, to a whale. So it seems like the connections between these neurons sort of allow us to have the cognitive abilities that we show, in terms of abstract thinking, for example.
Wow, I'm super interested and excited about your talk today. Maybe we can dive right in.
Yeah, so I'll share my screen and I'll start. So, great. So I think, can you guys see my screen okay?
Yeah, I think so. Sorry, I forgot to announce to the guests today that you guys can always interrupt and ask questions. And there's also an option to just type the question in chat. I'll be monitoring chat and reciting the questions. If you want to do that, that's fine too.
Yeah, absolutely. Please feel free to ask questions in the chat or just unmute yourself. Um, so thanks for introducing me, Prachi. And yeah, my name is Sergio. I'm a graduate student at Columbia. And I hope that in today's presentation, I can sort of bring light and make you aware of just certain aspects that make our brain different from other animals. But I don't want to undermine, and make it seem like our brains are so vastly different. Many parts of our brain are pretty much similar throughout the animal kingdom. And this is true for many animals. And a lot of them show many complex behaviors that we can't even begin to understand. Um, so so with that, let's just go into it. So just to show you sort of where we are and where the human brain is sitting, I thought I'd put this picture here. So this is sort of a translucent image of looking through, for example, if we saw no bone or skin, where the brain is sitting. And we also see a lot of these fibers coming out of the brain, for example, innervating the skin of your face. And all of these neurons are responsible, and fibers are responsible for getting input from the outside world and letting you know what's going on. So this is how, for example, you know whether your face is wet, you can feel wind going across your face, you know when something is touching your face. And this is because all of these fibers are making connections to the surface of your skin and translating any input that they get into your brain, and then it's getting processed. And I think it's important also to realize that your brain is very much interconnected with your entire body. And that's shown here. And this was shown, this is an image from a museum where this is real, and they took somebody who donated their body for science. They took out all of they took out all of the skin, bones, muscles, and then just sort of fixed in place their brain and all the connections to their brain and their neurons. So here you're actually just seeing neurons and their nerve fibers, even along the spinal cord. There is no bone. So I think this really puts into perspective that your brain is very much connected to your entire body. So you shouldn't see it as just the brain sitting on top of your head as a separate organ. It's an organ that is interconnected to every other organ in your body. And it's made up of cells, just like every other organ in your body too. And we've just termed them neurons. So just to sort of compare our brain to brains of other animals, and this is to scale. So for example, here you have the brain of an elephant, much bigger than ours, which is here the brain of a human. Here you have the brain of a dolphin, the brain of a zebra. And you can see that our brain relative to an elephant is a little bit smaller, but it's a little bit bigger than a dolphin's and a zebra. And if we compare it to even other animals, here you can see the brain of a manatee, a chimpanzee, gorilla, pig, rabbit, and all the way down here, super small, is the one of a mouse. And what I want you to take away, hopefully, from this image is that despite all of these brains having different sizes, what we couldn't understand for a very long time was, why is our brain so different from that of an elephant, for example, despite it being smaller than that of an elephant? And what we're finding nowadays is that using the size of a brain is a wrong way to sort of correlate whether or not we are smarter or more intellectual. So we're moving away from using brain size, and I'll show you later what we're using nowadays to measure our cognitive abilities. But brain size is not a good measure because an elephant inherently will have many more neurons than a human does. But we can't seem to find sort of abstract thinking, complex language forming, and such things like that in the elephant community, for example. So yeah, so that's all I want to say about brain size. Don't think about it in terms of brain size. So now if we sort of zoom in into the microscopic scale of these brains and we think about, well, what are they made up of? And they're made up of these cells that we've termed neurons. And these neurons, we've turned them and we've categorized them based on their expression of DNA. So we know that your entire body shares all the same genetic information, but each organ within your body will express that information differently. So all the cells in your brain have chosen to express specific portions of your DNA that help make neurons. And some of these neurons, if we look at them, you have many different types and they all look very different from each other. So for example, in this case, you could have a sensory neuron that looks like this. This would be its cell body, where we have the nucleus containing its DNA information. And that information is driving the morphology or how this neuron looks. If we look at another one in the brain, like a motor neuron that connects your brain to your muscles, for example, so that you can move, it sort of has more of this shape. If we look at a pyramidal neuron, it looks highly branched. It seems to have, it almost looks like a tree. And these neurons are very important in your brain to connect neurons to each other and help them communicate and transfer information as your brain is trying to process it. So we see that where they're highly branched, almost like a tree. And then your brain too has its own immune cells responsible for clearing anything that it thinks is garbage and helping keep your brain in a clean state. And two of these neuron types are called astrocytes and microglia. And you can see that they're very, very, very branched. And this helps them move in between all of your neurons, clear out garbage, and helps maintain a healthy brain. So hopefully you can see that your brain is made up of neurons, but within these neurons, they are very different from each other, and each one has a very specific role to fill in your brain. So if we sort of zoom in terms of what are these neurons really doing in your brain? So let's take, for example, just one neuron, which we see here. And if we sort of let's connect it to another neuron downstream of it, right? So here we have two neurons, and they are connected by something that we call a synapse. And this is a very, very, very small connection here that allows this neuron to fire an electricity basically to the end of it. And then using chemicals, it will communicate with another neuron across its synapse. So these synapses are very important because one neuron can make many connections to another neuron. And we know that if those connections aren't properly maintained, then you can lead to many disease types. So let's now take, for example, something that would happen very simplistic within your body. So let's add another neuron to this set, this one here. And this neuron is responsible for telling you to raise your left hand. So whenever this neuron fires, it's supposed to transmit its signal down all the way to this neuron down here. And you should be able to raise your left hand, very simply. Now let's say there's a different neuron, not in this sequence, over here connected to a new set of neurons. And this one is responsible for telling you to raise your right hand. So whenever this one fires, it'll propagate its message all the way down, and you'll raise your right hand. But one thing that we run into a problem very quickly, and this is where we try to sort of understand what's going on in your brain, is what happens if two neurons, one that's telling you to raise your left hand and one that's telling you to raise your right hand, are both connecting to the same neuron. Now this neuron here needs to decide, well, which one do I listen to? Let's say you don't want to raise both of your hands at the same time. You only want to raise one. This is very complicated because this neuron here in this sequence, this middle one, oh, I'm sorry, this middle one here. Oh, sorry guys, just trying to use the drawing. So this middle one here needs to decide which input from each of these do I listen to? Do I listen to the one that says raise your left hand, or do I listen to the one that says raise my right hand? And this is very complicated, and I don't have an answer for you guys. And this is something that we're actively still trying to research in many scientific communities and research labs. So in my lab specifically, we study this connection, and I'm going to speak heavily on what's going on in a synapse. So what is going on in this little connection here when one neuron connects to another so that a signal can travel all the way down and that you can have some sort of behavior output. So you can raise your hand, you can walk, you can eat, you can talk, you can do many different things. So one thing I sort of wanted to ask you guys is, you can write it in the chat, but I wanted to ask you how many, let me clear this. So how many individual neurons do you think a human brain has? And you can guess. Do you think it has 100? Do you think it has 1,000, 1 million? How many do you guys think it has?
All right, so I'm just going to recite all the guesses in the chat.
Yeah, go for it.
We have 10,000, we have 100 billion, we have 50,000. Anyone else want to take a guess?
Good guesses.
No one knows.
Yeah, it's a hard question. So I'll tell you the answer. Great guesses. So the answer to how many neurons a human brain has is it has around 100 billion neurons. And this is sort of a hard number to understand, but sort of to give you a little bit of perspective, we know from previous studies that a lot of people have spent time on that this is roughly around the same number of stars that you could find alone in our Milky Way galaxy. So if you counted all the stars in our galaxy, they would be roughly around 100 billion. And this is how many individual neurons your brain has. And remember that all of these 100 billion neurons are all connected to each other, and they are constantly talking to each other. And they need to parse out a lot of information about the world around you, and information about your actions that you want to do. So this is very complicated. But going back to our theme of brain size is not correlated to sort of cognitive abilities. If we look at how many neurons a whale has, they have roughly around 150 billion neurons. So they have many more neurons than us. So there are animals that have more, and there are animals that have less neurons than us. But in either case, this is a lot of neurons that are compacted into a very, very, very small space inside your head.
Sergio, can I ask a question?
Yeah.
How do you count like how many neurons we have in our brain, or like what for any species? Like that's not something I imagine. You obviously can't count that high of a number.
Yeah,
Is it just calculated, or?
That's a really good question. And actually, I was reading up on it recently. Somebody did a study where if you actually went in and individually counted the neurons in our brain, somebody said it would take you roughly around 3,000 years to complete that count. So we don't do that. So the way we count it is we take a brain and we take a very small portion of it and we count all the neurons in that little space. And then we say, okay, well, we know the dimensions, we know the length, width, and height of this, and we know there's 100 neurons here. So we can just sort of multiply out by the volume of the brain and assume that. And that's actually how astronomers also count the number of stars. We don't, we're just assuming that there's a small portion in the Milky Way, we count it, and then we extrapolate out and say, this is how many we think there are. Yeah, but great question. So if it's not the number of neurons in our brain that makes us different, because we know there are animals that have more than us, then scientists started to sort of ponder and think about, well, then where are the differences then? Why are we able to have ideas? We're able to communicate them. We have complex language. We can write ideas through history, and we can learn really well, really fast when compared to other animals. So something that scientists started to think about was, let's go back and take our neuron, for example, here. If we take that one neuron and we start sort of asking, well, what are the number of connections that that neuron makes? And let's compare that between different species of animals. So we're going to compare the number of connections between different species. And let's say we're going to just say, if it's very high up on the axis, these are a lot of connections, or we have very low connections. So if we plot this and we look at different animals, let's say a bird would be somewhere around here. It would still has a lot of neurons, but the connectivity in its brain is very low. So for example, one neuron may connect to only 3 other neurons. So it's very low. If we move on and we say, well, how about a mouse? It has a little bit more connections than a bird, but not that many more. So maybe one neuron of it has maybe 10 connections. And if we look at a whale and a dolphin, whose brains are actually very much larger than a mouse, they actually all roughly have the same number of connections between their neurons. So even though the brain is very vastly different in size, the connections that their neurons are making are roughly the same. So we're now sort of asking, okay, well, maybe there's something to learn here. Maybe it's the connectivity in our brain that makes us unique. And that seems to hold true. So when we look at one neuron in a human brain and we ask how many connections it's making so many more than any other animal species that we can look at. So that means that maybe one neuron in a human brain is making maybe 1,000 connections to another neuron. And that makes it very difficult to study, but it also makes it an interesting question, because this is maybe the difference where the difference in our brains, basically. It's not in the size, but in the connections that those neurons are making.
That's so interesting.
So
Can I ask, oh, Sergio,
yeah,
does that involve like just more projections like of the what is it, the axon, like reaching out and touching different parts of the next neuron? So
yeah,
okay, wow.
exactly. So, and I'm going to show that later, but yeah, if we imagine and go back, I can show this in the next slide. So here we have, right, two neurons. This is their synapse to their connections. You can imagine that it's not only just making one connection. So this neuron with its axon here is making maybe 100 connections to the next neuron. So yes, it's in the terms of projections that it's making across a synapse. And it's here where we're finding the vast majority of differences between humans and other animals. So it seems like one human neuron has 1,000 connections to only one other neuron. But if we start then sort of multiplying out the numbers, we notice this gets really complex because I've told you, you have 100 billion neurons, and each one of those 100 billion makes 1,000 connections. So you can see that the numbers become sort of skyrocketed if you were to sort of count the number of synapses, which at the moment is almost virtually impossible for us to count that.
That's
Sorry,
crazy.
I had a follow-up question. Like, is this
Yeah.
dependent on which area of brain you're looking at?
Yeah, so it's very dependent on the area that you're looking at. So most of the connections that you make are in the areas where you're processing sensory information. So whether that be visual or olfactory smell or touch. So in areas where you have to process the sensory stimulus from your external environment, you find the most number of connections. You actually find a little bit less in motor areas involved with moving. Despite that actually being one of the most costly things that your brain has to do. So two of the things that are most costly to your brain are maintaining all the neurons firing, but also moving. That's actually where most of your metabolic energy goes into.
One more question based on that. So if we compare our abilities to some animals in terms of sensing, some animals are better than us. Like, for example, dogs can hear more than humans can. So in those areas, are their brains having more connections than humans?
That's a great question. I'm actually not sure. What I would say is that yes, there are other animals that are highly specialized and highly sensitive to detecting stimulus. For example, your dog can detect smells really well, but it then has to give up other senses. So maybe your animal can smell really well, but dogs actually can't maybe see very as well as us. So humans seem to be an outlier in that all of our senses are highly attuned versus other animals have chosen to have one specific highly attuned sense. Yeah, but it's a great question. I don't know if you looked at a dog, for example, maybe it has more connections in parts of its brain involved with smell. Yeah.
Yeah, and maybe like some frontal areas don't have as many connections as humans do, for instance,
For
because
instance, yeah, since yeah, we have more sort of problem solving abilities that are more found in our frontal lobe areas. Yeah. So yeah, so again, we're focusing in on how these neurons connect to each other. This is where we're finding the differences. And just to give you a perspective of how fast things are happening, when a signal comes from this neuron and travels down to this one, it's traveling at roughly 150 miles per hour. So when your neuron fires and sends a signal to another neuron, if you were to measure the speed of that signal, it would be on average 150 miles per hour. Now, this is really fast when you think about how small these neurons are. So these signals are moving incredibly fast between regions in your brain. And there are so many, and yet you're able to function really well. And just to give you a perspective of what we see in the lab, when we look at a neuron and look at its connectivity, the next slide, here I want to show you, let's again zoom in on one neuron. And in this neuron in the lab, we're going to label every single point that this single neuron makes a connection with to another neuron. So what we see here is this is actually what we see in the lab. This is a real mouse neuron. We've labeled it with red. And every single little green dot that you see on this neuron is a connection that it has made to another neuron. Now, this is a lot of connections that this neuron needs to deal with, right? I only showed you in the diagram before one connection, but now this neuron here needs to really parse out many different inputs into it and decide, well, which one am I going to listen to? Maybe this neuron is connecting to a region in your brain that is involved with visual processing. Maybe it's also connected to a region involved with movement. Maybe it's also connected to a region involved with smell. So we see the complexity here. And this is a mouse neuron. So this is actually not even the most number of connections that we could possibly see. If we were to stain a human neuron, we would see that these dots or synapses would just skyrocket a lot more along this neuron. So again, sticking with the theme of connectivity, our lab wanted to understand what are the implications of having more or less of these connections, both at the microscopic scale, but also in terms of a behavioral output. So if you have more or less, does that affect the architecture of your brain? And if you have more or less synapses, does that affect the behavior of an animal too? And to understand this, we sort of took an approach where we wanted to understand the evolutionary scale of things. And we wanted to compare our brain to all the previous iterations of brains that came before us. So if we look at a sort of this evolutionary tree here, it's super complex. You can see all the different species that have evolved over time. It starts at around 4 million years ago. And then all the way over here in this tiny little section, we see the evolution of humans. But we notice that if we think about it in the context of this evolutionary tree, we notice that our brain had to go through many trial and errors in order to get to where we are today. So maybe by understanding the previous brains of previous evolutionary animals, such as a chimpanzee, a gorilla, even a monkey, or sorry, monkeys in general, or maybe even a mouse or a hamster, then we can start getting closer to how did our brains evolve in terms of connectivity to what they look like today. And if we sort of zoom in on this evolutionary tree and just focus on, well, what was the evolution of brains look like? And we sort of get this map. And you can see a bunch of different animals here, humans all the way here on the top. And we see that our brains are pretty similar to a chimpanzee's. And every colored region here means that it's similar between species. So if we look at a squirrel here, its little red area in the brain here is very similar to our red region area in a human. So we notice that regions within our brains are conserved across species. So it's not that we are changing the function of regions in our brain, but it seems like we are changing the connectivity between regions in our brain. And this sort of led to many scientists ask questions where, well, where can we find these differences? If it's very hard to just find them at the anatomical scale if you're just looking at a whole brain. So where can we look for them? And scientists decided to start looking at differences in DNA to begin with. So we know that every cell, we can extrapolate DNA from it and we can compare it. So let's say, for example, scientists took human DNA, they took, chimpanzee DNA, and then they also took mouse DNA, shown here, right? So let's say I have all the human DNA laid out in a string. I have all the DNA from a chimpanzee and all the DNA from a mouse. Now, what I'm going to find is if I look for similarities between those pieces of DNA, I'm going to actually find many similarities. So if I put a blue bar over here, I'll notice that all these regions of the DNA across these species are the same. So all of these match up, all of these match up, and all of these match up. And our DNA between a human and a chimpanzee is very similar, on average, somewhere between 90%. But actually the DNA between you and a banana is also pretty similar, which is roughly around 50%. So we're not looking for the similarities. What we wanted to look for was the differences. So when scientists started to look for human specific differences, so we went into the human DNA and we asked, what portions of the human DNA are not found in any other species? And we focus on the DNA that is expressed in your brain. So let's say, for example, in a study, we found many of these regions, let's say, for example, here in red, that this piece of DNA containing a gene is not found in any other animal species. And we know that this was a region expressed in your brain. So this makes it a good candidate to test whether or not it carries functional implications. So what we can do is, and what scientists have done, is they'll take this piece of human DNA, they've identified this little difference here that is, again, not found in any other species. What we can do then is we can take molecular scissors, we can cut out that little piece of DNA, and now I only have that little piece of human DNA that contains one gene. So I'm going to focus on studying that little piece of human DNA that has a gene that I cannot find anywhere else. And I know that it was specific to the brain. So what I can do now is I can take that little piece of DNA, I can take a cell, a neuron that is not human, and I can introduce that little piece of human gene that I cut out, and I can put it into that non-human neuron. What that allows me to do is I can now ask, well, when I introduce that little human gene into a neuron that is not from a human, does that maybe change the connections that that neuron is now making? So what we've done now is, right, we have a way of expressing and seeing changes of a human specific gene in a non-human neuron. So we now started asking in my lab, well, let's see if we could sort of drive the same connectivity that we see in humans in non-human neurons. Can we increase the connections that neurons make using some of these genes that were found in the brain that were only specific to humans? So if we go back and take, for example, our neuron, let's say this is a neuron that we haven't manipulated, it doesn't have any gene inserted into it, and we just count the number of connections that it makes. So let's say we count them and we have this gene and it's making 2 connections so far here. So we can count them. We know we have 2 previous connections to 2 neurons. We can go in and we can say, okay, it's making 100 connections here, 100 connections here. We can study it all we want. But now what we can do is with that same neuron, we can decide to, at a very specific time point, express our human specific gene, which, remember, we cut out from our long piece of human DNA. So now what we can do is we'll bring in that piece of DNA that we had cut out, we'll introduce it into our neuron, and now let's ask, does that single piece of DNA we put back in increase the connectivity of this neuron and sort of mimic what we see in a human brain? And that actually seems to be the case, and we can do that with just one neuron. So when we express this one neuron, we now see that it's going to make a lot of more connections. And this was a really, really outstanding sort of discovery that we weren't expecting in the lab. We weren't expecting just, for one single gene to mimic the connectivity that we see in the human brain. And this had really a lot of implications because now we had a way of probing and making a non-human brain seem more human. And that helps us understand not only connectivity, but also helps us understand more how diseases may progress and how also we can treat them. But it was remarkable that just using a single piece of DNA containing one single gene could drive many more connections in a non-human neuron.
I have a question.
Yeah.
So does this mean that intelligence is directly related to your genes? And even in humans, can you compare the gene expression and correlate it with the intelligence?
Yeah, so it's a great question. And we get it a lot, especially in my lab with trying to correlate intelligence. I wouldn't correlate intelligence to your gene specifically. I can't comment on intelligence, but in terms of connectivity and sensory processing. So I think what we're finding is when we do this in a mouse, for example, so if we take this little piece of DNA and we put it into some neurons in a mouse's brain, and we let those neurons form connections that are sort of similar to humans in the term of number of connections. It seems like those mice do learn tasks significantly faster. They are much more sensitive to stimuli. But this is all to say that it seems to be more correlated to connectivity. So therefore, it seems like while you drive more connections in your neurons, then they are better able to communicate with each other. So they're able to parse out stimuli better. That's not to say that the mice are more intelligent. They're just more sensitive now. So if you put this, let's say, for example, in a region in their eye, maybe they're seeing better. Or if you put it in a region in their brain that is where they process stimuli from their whiskers, and that's actually what we do, they seem to be very sensitive now when they use their whiskers to feel their environment around.
And has anyone studied the connectivity relationship with genes in just different types of human population?
No, so as of right now, I don't know of any study that has been done in. One of the problems that we have is that this gene that I'm showing you here that we study, it's called, and you can look it up, it's called SRGAP, SRGAP. One of the things that makes it hard is that, again, it's a gene that's only found in humans, and we can't really do much experimentation on humans. So it makes it very hard to understand what this gene is doing. And that's really why we cut it out from a piece of human DNA and put it back into a non-human neuron to sort of try and answer these questions. But it makes it very hard, yeah, to study within humans at the moment. Yeah, so we sort of have to correlate what is going on maybe behaviorally in a mouse model.
That makes sense. Thank you.
Yeah. So this was really, really an outstanding scientific discovery that we sort of found in the lab. Again, this single gene could drive many, many more connections in your brain. And something sort of along the lines maybe that Prachi was asking, it's not that you're making more connections with every neuron in your brain. They're actually making connections to the normal places that they would. So what I mean by that is, if you have a neuron in your visual cortex, for example, all the way in the back of your brain, and it makes a connection to somewhere in the middle of your brain. If we put this gene back in that visual stimulus neuron, it won't just make random connections, it will simply make more connections to that middle region of your brain. So the connectivity across regions is maintained, but you have more connectivity across those regions. And again, I'll show you, and I want to bring up this picture again, that this is very hard for us to understand, because here I'll show you an image and it sort of rotates again, this one neuron where every single little green dot, you're seeing a connection to another neuron. So again, it's almost counterintuitive that maybe you don't want to drive more connections in this neuron, right? Why do you want to make this neuron more complicated? It's already as complicated as it seems it's going to get. But it seems like more connectivity leads to better sensory processing. And something that I'm not showing you here, which we have to think about, is that many of these connections you can think of are either excitatory. So a connection can be excitatory, and that neuron can excite the next neuron. But many connections are also inhibitory. So you can have a neuron that signals and tells the following neuron not to fire. And you have a very specific balance between excitatory and inhibitory neurons in your brain. And adding more connections means that you need to be very careful with which connections are you making more with. Do you want to make more connections with your excitatory neurons, or do you want to make more connections with your inhibitory neurons? You really want to maintain that balance, and you need to be careful. Something too that we're finding is that the inhibitory neurons in your brain are actually a little bit more important than the excitatory, because those are able to sort of bring down the activity in your brain and keep less neurons from firing, because you have already so many of them. And I want to show you a video, which is this was taken from a zebrafish, a very small fish. And you can image its brain in real time. You can sort of see all the neurons that would fire. So let me see if I can play this. I'll play it from the beginning. Let's see if I can increase the quality for you guys. Well, I'll let it play. So here we're watching a fish, and you can see that every single region that you're seeing light up in red are actually groups of neurons. You can see in the front that there is a huge amount of stimulus, and that's because the zebrafish is being presented in real time a stimulus, which is these graded sort of black and white bars, which are not so important. But what I want you to take away is these neurons are firing and connecting to many different regions within your brain, and they're having to parse out all this information. And what's really cool about this is that because we can image in real time a live brain and its firing, what we can do now is we can reintroduce that piece of human gene that I showed you that we've clipped out, put it into some set of neurons, and then we can use this technique and ask, well, now when we have more connections using our human gene, do we change the way that these neurons communicate with each other? And we can see that in real time. We don't have to ask static questions. We can ask real-time dynamic questions of what these neurons are doing. So it's a very powerful tool to take. We've gone from one gene and correlated it to the macro population scale of how neurons communicate and where are the implications of having more of those connections. So sort of to wrap up too is, well, this is all great, but why should we really care about all of this? Why are scientists spending so much time in trying to understand the connections between neurons in your brain? One of the things is just scientific curiosity. We want to understand things of how memories form, how thoughts and ideas arise in our brain. And these are very complex. And as of right now, most of our models, right, we can't ask the animals to tell us what they are thinking or how they are forming memories. And that makes it really hard for us to understand what changes are we driving in them. So maybe by understanding a little bit more about the connectivities using other animals, we can understand about how those connections implicated in our memories. Another reason that we really do this in the lab, in my lab specifically, is trying to understand these two diseases, which are Alzheimer's disease and autism spectrum disorder. And both of these diseases are really detrimental. Maybe you guys also have family members who are suffering from these diseases. And we really want to find ways that we can treat them. And in this sense, in Alzheimer's, we notice that we not only have a loss in the number of neurons, but we have a loss in the number of connections that those neurons make. So maybe by understanding this one gene that makes more connections, we can sort of exploit it and use it to treat Alzheimer's, to drive and keep more connections going. And maybe we can not have any of the symptoms associated with it. Autism spectrum disorder is another disease that we are trying to also understand in our lab. And this is sort of a disease that is implicated in, you have a very critical period window when you are a young child. So anywhere between when you are born to maybe around, I'd say around 20 years of age, your brain has to triple or quadruple in size roughly. That means that you're not only making more neurons between that time, but you have to establish a lot of new connections. And those connections are established by trial and error. And that means trial and error in terms of you interacting with the outside world. So the more you move, you learn, for example, how to ride a bike. In the beginning, you might fall many times, but after a while, you create strengthening the connections between neurons that help you to ride that bike. And in a disease like autism spectrum disorder, we see that this critical period window is closed very early. So these children in a very young age solidify the connections that they have in their neurons in their brain, and they don't allow for new ones to form. And that carries on to a lot of behavioral deficits in the long term that we would like to understand and sort of treat and be able to treat them better to help. And with that, yeah, I would just like to end. Thank you for listening. And really, if you have any question, feel free to ask it. Yeah, thank you for listening.
Thank you.
Thanks, Sergio.
Yeah.
Yeah, I have a question. So in terms of expressing that gene, that increased connectivity, is is there a way to selectively express it in inhibitory neurons instead of excitatory neurons? And do you see like sort of a lessened state of like awareness or like behavioral changes for a mouse?
Yep, that's a really, really great question. So that's actually something that we're doing now. So right now, very recently, we found a tool where we can inject a piece of DNA into a single neuron. So we can take a real live brain, we can drive sort of a needle, and that needle will touch one neuron, and then you can deliver DNA to just that single one. So you can select different populations of your neurons. So for example, as you said, you could select an inhibitory neuron. You can put that piece of DNA back in and see what happens when I drive more connections in that one. Do I see an effect? And what's important is that we're trying to take both approaches. So we can drive connectivity in the whole brain, right, by just introducing that piece of DNA into every cell, but then we don't really know what's going on. So what we're trying to understand now is by putting it into one neuron, what's happening there and sort of working from one neuron to many more. But as you saw, we have a lot of neurons to work with, and we can't put spend so much time putting it into each one. But going back to the inhibitory question, as of right now, the gene actually seems to predominantly increase connectivity in excitatory neurons, not in inhibitory neurons. We're not sure why that is, but we're trying to sort of understand that now.
That's really interesting. Thanks.
Yeah.
Feel free to ask questions here or in chat.
Let's see. So does your DNA indicate?
I can decide maybe. Does your DNA indicate or have connection with how neurons are developed in your children?
Yeah, that is a great question. So up to a certain point, yes. So for example, this gene that I've been talking to you about that is found only in humans, we found now that this gene can have certain mutations in it that doesn't allow it to be expressed properly, this SR gap gene that I was talking about. And when you find those mutations in that gene, it's usually implicated in autism spectrum disorder. And that's again, because when you don't have this gene I've been talking about to allow more connections to form, you really close that critical period window where you want children as they're growing up through early adulthood, even into their mid-20s, early 30s, to have a lot of what we call synaptic plasticity. And the synaptic plasticity is what allows you to create new connections, create new memories, create new behaviors and such. But mutations, at least in this neuron, sort of close the potential of those neurons to make more connections. And that has deleterious effects, such as autism spectrum disorder. So up to a point, yes, your DNA does influence how the connectivity in your brain will develop.
How is that gene you reference spelled again, please?
Yeah, it's SR gap 2 here.
Sergio, do you know these commercial gene tests that are available are capable of detecting things like these?
Um, wow, that's a great question. I am not sure if they can detect mutations in the gene I am talking about, but I do want to maybe I want to clarify because it seems like there's more concern of this gene now. So just to clarify, the gene is sort of it's found across all humans. So the the mutation rate in it is still very low. And I want to make clear that this is just one gene. And this one gene may actually need many other genes to be mutated to actually have a deleterious effect. So you shouldn't view it as, oh, I have a mutation in this SRGAP2 gene. I won't make the proper connectivity that I need. That is likely not the case. You need an entire set of repertoire of genes and proteins to communicate with each other for proper development. We've just found that this one gene causes more connectivity, but that isn't to say that we're not causing deleterious effects when we drive more connectivity. So you also have to think about that. So when we put this gene, for example, back into a mouse, and we say, oh, it's learning better, it's having better tasks, but we actually don't know what sort of damage we may be doing to it. Maybe it's not processing other information properly. And maybe that's why evolution decided to not have that gene in other animal species, right? So just because it works in us doesn't mean that it has to work in other animals. But that's sort of the questions that we're trying to understand by using it. So you can understand the default state of the system, but to understand it, you need to perturb it and you need to sort of ask, well, what goes wrong? And by understanding what goes wrong, we have a better understanding of not only how we can treat those wrong things, but how is it the proper development of the brain happening?
Do you think that in future medicine will involve just gene injections to fix?
Yeah, who knows? And I know, I know there was a study recently, or it came out where sickle cell anemia, which is actually uh, if you have a protein that's made up of amino acids, and sickle cell anemia is just one change in one amino acid on that protein.
Oh, is this? I remember there's something
Yeah,
beta gene, alpha gene, something was messed up in that disease. Yeah.
yeah. So one way that they were trying to treat it was by sort of injecting back into that individual the correct form of that gene. Um, I don't remember whether or not it was successful, but I know that we are trying it. Um, but of course, this is all great when you think about disease. This is great when we want to treat Alzheimer's, or we want to treat autism spectrum disorder or Parkinson's and things like that. But you always have to keep in mind and be cautious that once you're injecting DNA back in, you sort of want to also keep in mind the ethical implications of you injecting that DNA back in. You should always think about those. So it's not just for treating disease, but you can always have people that maybe ask, well, I want to have bigger muscles, or I want to be stronger or better, and things like that. And I think we shouldn't think about it in that way. We should really stick to how can we use these findings to help those that have really deleterious medical problems.
Thank you so much. Uh, does anyone else have any questions?
Ooh, I have a small question. So the photo was amazing of like all the different green synapse dots.
Yeah, I can go back to it and put it while you ask your
Yeah.
question. Go for it.
Does it make a difference really, like how close a connection, like a synapse is to the cell body versus like way at the ends of the arms, as to like, will that like help break the threshold to cause a signal to go down better or worse if it's like closer to the actual core of the cell?
Yeah, that is a really good question. So yes, absolutely. So if you make a connection, for example, let's say in this neuron, if you make a connection all the way up here, then that input that it's getting actually has to travel much longer, because something I didn't speak about, which you are getting at, is once you get, for example, let's say a neuron that fires at this synapse up here, and you get a little action potential here, that message needs to travel all the way down to the cell body. And it's here where that sort of message accumulates at something we call the axon hillock. But it accumulates, and basically there it's where the neuron decides, will I transmit that message or will I not? And something interesting is that many actually excitatory inputs are far away from the cell body, and more inhibitory inputs are near the cell body. So when you make those inhibitory neurons are actually inhibiting that neuron from firing, and it's actually making those connections very close to where the neuron needs to decide whether or not it will fire.
So the excitatory ones have to be especially strong to get past sort of
Have to be exceptionally strong. And that's why we actually see a little bit more excitatory neurons than inhibitory in your brain. But your inhibitory ones seem to carry more weight, if you want to call it, because they are so close to the cell body where that neuron needs to decide, will I fire or not?
That's so cool. Thanks.
Yeah.
We have some questions in the chat. How do we create more neurons in our body, and does exercise create new neurons in your brain?
Yeah, great two questions. So do we create more neurons in our body? Yes, but it is in very specific regions. So in your brain, you can't really find more neurons maybe once you get past a certain age, usually adulthood, well into your 30s. But there are regions in your brain that you actually don't think about, that are part of your brain, that are constantly creating new neurons, which is your olfactory system or your sense of smell. So those neurons, every time you blow your nose or you get sick, they die, and you get rid of them. But you always can smell and you regain them back. So that's one area where neurons actually repopulate constantly and are keeping you going. When you get more into the central brain or cortex regions, you don't find necessarily that you can make more neurons, but what you are constantly making is new connections and pruning away old ones. Exercise, in a way, actually promotes not new neurons to form, but new connections to form. And that's sort of, again, the theme that I wanted to carry here, that it's really these connections that are allowing you to process information better. And yes, exercise has been shown to increase the number of connections that you make between neurons. And that has sort of health benefits. One study that I can tell you quickly about was, there was a study where they were looking at why certain people, when you look at a brain that has Alzheimer's disease and compare it to a brain that doesn't, one that has Alzheimer's has many less neurons in it, and they die and they have many less connections. And that leads to a lot of memory loss. But there are certain people that never reported having any symptoms of Alzheimer's, yet when they would donate their bodies to science, their brains looked exactly the same as a brain that had Alzheimer's. So scientists began asking, well, what does that mean? How come some people show the same morphology as somebody who has Alzheimer's, but don't show memory loss? And what they found was that many of those individuals, they didn't know it at the time, were actually nuns. And these nuns were spending a lot of their time reading. And just constant reading was causing them to make more connections between their brains. So despite them losing neurons, they were counterbalancing that by making more connections with the neurons that they had, even though they were less. So you can help sort of not get some of these phenotypes or these behavioral or disease phenotypes that we see by helping drive more of these connections. And learning is also involved in that. So when you learn something new, you make a lot of more new connections in your brain.
Any more questions? we answered everything. Thank you so much, Sergio,
Yeah.
for joining us
Thank
today.
you, Sergio.
Thank
Yeah.
you.
That's great.
Awesome.
Fine. Bye, guys.
All right, so we can end the session here. Thank you so much for coming, everyone. And we hope to see you next time also.
Thank you.
Yeah. Thanks for coming.
Thank you.